Hello, Assalamu alaikum. Uh, Assalamu alaikum. This is Science for All channel. For today's video, our topic is DNA replication. We will discuss about first of all the definition, what is DNA replication. Then we will move towards different models that are proposed by the scientists for the DNA replication. So, starting from what is DNA replication, as you have heard the word of replication many times, replication simply means a copy, as you hear the word replica. Similarly, DNA replication is basically the copy of DNA. So it is the biological process we say because it occurs in living organisms, and in this process copies of DNA are formed. We can say it as DNA duplication or DNA synthesis. And what's the time the DNA replication occurs? So it basically occurs at the time of cellular division, which we call as mitosis or meiosis, particularly in the S phase, which we call as synthesis phase of mitosis and meiosis. We will discuss about these in our next on videos, what is mitosis and meiosis and other phases. So for this time, you can say you can remind that DNA replication occurs in the S phase of cellular division. So yes, uh, how much time is required for the complete replication of DNA? So it is found that human chromosomes contain about 150 million base pairs, so it is a very huge amount. And since cells are working all the time, so cell replicates almost at the rate of 50 base pair per second. So a second is required for replicating 50 base pairs. So if the cell continues with this speed, it almost take a month to copy a chromosome. So it's a very huge time. But there is a process. You can say for the compensation, there are multiple origins of replication within the cell, within the chromosome. So it is not like there is only single point of replication in the whole chromosome. So there are multiple points of replication. So when the replication at all of these points are completed, so we can say the whole chromosome is duplicated. So these two strands are joined together between these base. We call it as a base pair. So we can say that adenine is hydrogen bonded with thymine and guanine is hydrogen bonded with cytosine. There are always two hydrogen bonds between adenine and thymine, and guanine and cytosine are joined together by three bonds. So we call it as complementary base pairing, that adenine will always bond with thymine and guanine with cytosine. So yes. Models for DNA replication. Three models are proposed by the scientists for DNA replication. One is semi-conservative model, second is conservative model, and third one is dispersive model. So we will discuss about what are these models one by one, and we will see which model is the most acceptable model and for what reason. So starting from semi-conservative model for DNA replication, Yes, there is a spelling issue. Conservative model for DNA replication. Okay. First of all, yes, here is the picture. You can see here is the DNA. These are two strands. For the ease, I didn't show the coiling for the ease of understanding. These are two strands of DNA, and these are the base pair. Like here is mentioned, red is showing adenine, blue is thymine, yellow for guanine, and purple is showing cytosine. So these base pairs are in complementary manner arranged. So at the time of replication, during first round of replication, what happened in case of semi-conservative model, that two strands unwind. The two strands unwind, as you can see in our new dotted strand, only one strand is the parental strand. As you can see here, the one strand is the parental strand. While on the other hand, we have a new daughter strand that is shown in green color. Here you can see. So parental strand is combined with the daughter strand in the daughter DNA molecules. So yes, this is the basic concept for the semi-conservative model of DNA replication, that the parental DNA unwinds, and when this unwinding begins, the daughter DNA strands begin to form along these unwinding DNA strands. So these parental strands act as template, and the daughter DNA strands are formed along these template strands. So the daughter molecules will contain a mixture of the new strand and the parental strand. So this is about the semi-conservative model of DNA replication. It's very simple explanation that I can give for the ease to understand this concept. Okay. So yes, we can move towards our next model, that is conservative model for DNA replication. As its name indicates, it says that parental DNA remain intact, completely intact. And after first round of replication, what happened that the parental DNA remain as it remained completely intact. There is no unwinding and nothing like this. So this model suggests that a complete new DNA molecule is formed, and there is no mixing over and other such things, as we discussed in semi-conservative model. So if we consider this concept, if this model, so we can say according to this model, the new daughter DNA molecule will be completely different from the parental DNA molecule. But what we see that generally what we see, the offsprings are more or less similar to their parents. They have some traits that are similar to their parents. But as far as this model is concerned, it suggests that a complete new DNA molecule is formed. So it will not contain any information like that of parents. So it means that the offspring will be completely different from the parent. So I think you can understand the disadvantages for considering this conservative model of DNA replication. We will discuss in detail about in comparison of these models in our next video. So yes, this is all about conservative model, that the parental DNA molecule will remain intact and a new daughter DNA molecule will be formed, and there will be no mixing over. So the last model about the DNA replication is the dispersive model. Dispersive means complete breakdown. So this model suggests at the time of replication, the parental DNA molecule is break down into pieces, and the daughter DNA molecule are formed in a complementary manner along these template pieces. Okay, along these parental or you can say template pieces, broken pieces of the parental DNA molecule, the daughter DNA molecules are formed. As you can see in the picture, that green is indicating the daughter strand and black is indicating the parental strand. So these are formed in complementary manner. So this dispersive model is more or less similar to semi-conservative model, But in case of semi-conservative model, we say that one strand remain completely intact. But in case of dispersive model, we say that the you can say the parental strand is completely break down, and daughter DNA molecules are formed in a complementary fashion along these broken pieces. So yes, these are the three models that are proposed by the scientists uh, for explaining the process of DNA replication. So yes, so here's the, you can say, comparison semi-conservative model, where one parental strand is intact and a new strand is formed in case of daughter DNA molecules. In case of conservative model, we say the parental strand remain completely intact and a new daughter DNA molecule is formed. While in dispersive model, the scientists suggested that the DNA molecule is break down into pieces, and the daughter DNA molecules are formed along these pieces in a complementary fashion, like adenine will bind with thymine and guanine will bind with cytosine. So this is very simple explanation without considering all other molecules and enzymes that are involved in this replication process. I've just provided you the explanation and understanding of these models that are proposed for the replication process. The question is, what do you think? Which model among all of these is the most acceptable model for DNA replication, and for what reasons? I think you may guess now which model will be considered as the most acceptable. So please comment your answers in the comment section.